There's a variety of impacts that harmful algal blooms have. We also have concerns in regard to these toxins building up during the bloom, and then where do they go? My name is Astrid Schnetzer. I'm an associate professor in marine earth and atmospheric sciences at North Carolina State University. I work on plankton food webs. My plankton ecology lab looks at microbial ecology in the sense of primary producers in different systems, both freshwater and marine environments. And we also look at how differences in plankton ecology affect ecosystem resources and ecosystem health. As part of our research, we're looking at algae or phytoplankton, which are the main primary producers in marine and freshwater systems. They're at the base of the food web, and they're extremely important for any sort of animal or organism in these environments. So if there is no algae or phytoplankton, you would also have no food for upper trophic levels. There's very good reasons to love phytoplankton, which we do in our lab. They're very diverse. They have all sorts of energy, nutrition strategies. They're beautiful, morphologically diverse, and typically extremely important, like I said before. However, there's a few of these algae that sometimes can bloom in excess and can cause trouble, especially if they're also capable of producing toxins. And these are called harmful algal blooms. There is a scientific consensus that harmful algal blooms are increasing in intensity, frequency, and magnitude globally. The standard answer from a scientific point of view is it's more complex, but there's also a point to be made that our ability to follow those algae that cause issues has become way more advanced. So we are monitoring more. We have better molecular and immunological approaches to look for these algae, but it is true that we have an increase in harmful algal blooms. So there's varying situations where you can find harmful algal blooms. Often they're associated with coastal environments where you have a lot of nutrients that make it into the coastal waters. So you can have nutrients coming from all sorts of sources. They can come from industrial sources, river runoff. They can come from sewage treatment plants. And these nutrients are true fodder for those little algae. And they take them up and they can divide extremely quickly. And that means they can build up their numbers from just a few per liter to millions per liter within a very short period of time. And that can overwhelm the system. So typically they're being fed upon, but in these cases, you don't have enough grazing and loss of that biomass. So you can actually see these algae, although they're microscopically small, they're smaller than like the thinnest hair on your head. You can see the whole water start to become colored because each of those little cells have a pigment inside of them. And if you have millions of them, they will start to show in the water. So you can see red coloration in freshwater, often green, neon green colors, and that's an indication of them blooming. There is different ways to approach this. One of the easiest ways to get an idea of what's going on with algal growth and activity is to look at pigments. Chlorophyll A is very easily measured and quickly determined in concentrations. But again, the problem here is that all algae, including cyanobacteria, have chlorophyll. So you will get an idea of growth activity, but not who's there or whether toxin is associated with it. However, it can be a first step to know where to focus in and what the spatial scale is. So satellite imagery can be an extremely helpful tool to figure out in which area you have these blooms and where they are distributed to, both in an ocean environment as well as in some of the freshwater systems. You can go and actually take a discrete sample, a water sample, and look under the microscope and try to identify which species are present. That gets you a little bit closer to knowing whether it's potentially toxic organisms. But in all reality, you do have to measure the toxin itself or some sort of expression of the toxin to really understand whether there is exposure risk.
So in Bogue Sound, we do sampling once every month. So we go down to the coast, we take water samples, nutrient samples, we collect water for phytoplankton counts as well as zooplankton counts, and we also collect spats. So we put the spats in the water, they stay there for a month, and we see what toxins are accumulating in the water. Right now, we're looking at microcystin as well as domoic acid. Microcystin is a freshwater toxin that's produced by a cyanobacteria, and domoic acid is a toxin produced by a diatom known as Pseudonychia.
So the Gulf of Mexico is a very interesting environment altogether because it's known for its strong anoxic events that are triggered by river runoff from the Mississippi and the huge delta. And it's also associated with algal growth and all that biomass degrading and then leading to anoxic conditions in the water column. If you go onto the Florida coast in regard to the Gulf, you might be familiar with the red tide, the Florida tide, which is a very severe type of harmful algal blooms because the toxins that are released there from a little dinoflagellate, a little microscopic organism, are actually also aerosolized, so they can't be released into the air. So if you have a bloom at the coast there, you have to stay away from the beach because our lungs are very efficient filters and it can affect people that way. There's a lot of research in each of those areas going on that tries to target the local problems. We're talking about marine algal blooms. Domoic acid is caused by a diatom, but domoic acid, once it's released into the food web or taken up through the food web, can cause amnesic shellfish poisoning. There's a lot of research on the California coast that shows that it might affect things like sea lions or pelicans. So if you have an algae that can bloom in coastal waters and affect a 400-pound sea lion, you have to start paying attention to that. And some of the studies have now started to use the newest technologies, starting with DNA-based detection methods to look at where in the water column and when can we find those algae, and have to be aware that there might be exposure.
For my research in the Arctic, there's the Alaska Ocean Observing System that created the Alaska Harmful Algal Bloom Network to kind of monitor changes in the environment, marine animals, and how they're affected by harmful algal species can bioaccumulate up the food chain, affecting everything from our filter feeders like our clams and oysters to our fish, all the way up to our marine mammals that may eat fish or krill or plankton, all the way up to humans can be affected by eating these animals or from the airborne toxins that can be produced from HABs.
I'm Dr. Talman, and I'm an assistant professor of shellfish pathology in the College of Veterinary Medicine. Most of my research right now deals with pathogens and disease that impact fisheries and aquaculture. So my lab works really close with industry here in the state regulators like the Division of Marine Fisheries. Most of the work right now deals with shellfish aquaculture. It seems to be the largest growing industry in the state. So a lot of the work that we do deals with disease impacts once you start culturing fish and shellfish on a really large scale. We are looking at shellfish aquaculture, shellfish in particular, as a sentinel for harmful algae, as a way to measure harmful algae. We know that toxin-producing algae are in coastal ecosystems, that they can accumulate, and that sometimes they can accumulate in shellfish. And so with expanding aquaculture, there's concern of human exposure to shellfish consumption. Down here in the lower part of the estuary, they receive much more flushing. We know their toxins can accumulate, but we really have no idea. One, whether they affect our species of concern at all, whether they're human exposure to shellfish consumption. So it's really a first cut, whether it's an actual issue.
Some of our studies take place in Jordan Lake, Falls Lake, in the Chowan River area, the Albemarle Sound. And in 2019, just before the pandemic hit, we actually we're able to document that some of the toxin levels here are really worrisome. There is recreational thresholds that are recommended to not pick anything out of the water, don't swim in it, and we were several orders of magnitude above those in the Upper Chowan River. Again, because these blooms are episodic, they happen very quickly, so you might easily miss them. They might be dispersed or rained out very quickly, so you really have to work with people in the area that can spot them, alert you to them, and help you document them well. And that was the benefit that we had in 2019. Another sort of thing that has showed up on everybody's radar is that there's multiple toxins that they might simultaneously occur in these environments, and then even if they might be typically in low concentrations, what does the combination of these toxins or continued low concentrations like chronic exposure mean for those who frequently consume these sources in these environments? So trying to find out what environmental conditions trigger toxin production is key. And for that, we need to get more and more data. We have to collaborate with people who do these studies all over. There's a lot of labs, there's a lot of information that can be consolidated to get a better idea. And we have to make sure that we go by region or environment, because what you find in the Albemarle Sound will be very different from what you have in Chesapeake Bay, will be very different from what you see on the Florida coast.
So I think as a field we've made really great strides in monitoring for these toxins and detecting that they're there. So we have kind of a better understanding of the scale of this problem and the severity in different regions. And I think moving forward, one of the biggest things that we're going to have to tackle is understanding the ecological and human health consequences of these different toxins and the different concentrations that they can exist at. I think there's some really interesting research going on in that sector of this field, and I'm interested to see what comes out of it.